Hello everyone, I'm Anas Al Ubaidi, and this is the second session or second lecture within Chapter 24 of the Advanced Medical Science Liaison and Medical Monitor Certification course program. As the chapter is about conceptual and theoretical monitoring, while the topic for the session is about monitoring of drug dimension and drug reality interaction trials. Introduction. Definition. Drug dimension interaction trials investigate the effectiveness and safety of drugs across varied therapeutic time points, doses, and populations under controlled settings. Drug reality interaction trials assess how drugs perform in real world conditions compared to ideal clinical settings. Significance. Identify variations in drug efficacy and safety between controlled clinical and uncontrolled real world environments, ensure broad applicability of therapy outcomes while safeguarding patient safety. Key points. We'll start with the protocol adherence. Strict adherence to trial protocols is essential to ensure consistency, data validity, and regulatory compliance. Monitoring for deviations is particularly crucial in drug dimension trials, where variations in procedure can distort findings around drug behavior under defined parameters. And here's a diagram summarizing protocol adherence in clinical trials. From left to right, formulate detailed protocols, communicate protocols, conduct monitoring, document deviations, investigate deviations, address deviations, retrain staff, monitor drug adherence, and then implement interventions. Best practices. Formulate detailed trial protocols and communicate them effectively to all trial sites. Conduct both on-site and remote monitoring to ensure procedures such as sample collection, timing, and drug administration are followed. Immediately document, investigate, and address any deviations. As for example, a dose escalation drug dimension trial faces a challenge when blood sample collection deviates from the scheduled window, resulting in reliable pharmacokinetic data. Monitors flag the issue and retrain site staff to ensure adherence moving forward. In drug reality trials, protocol adherence might also involve monitoring whether patients in everyday settings are taking the drug as prescribed. Non-adherence, when flagged early, can drive targeted interventions to improve study outcomes. Data integrity. Data forms the foundation of reliable trial results, ensuring its accuracy, completeness, and consistency is critical, particularly in drug dimension studies where large volumes of data under controlled settings are generated. And here is a summarized diagram on ensuring data integrity in drug trials. From 1 to 6, left to right, identify data integrity need, use real-time validation tools, employ centralized systems, cross-check reports, address anomaly example, and then monitor patient-reported outcomes. Strategies for effective data integrity monitoring: use real-time data validation tools to identify and correct anomalies immediately. Employ centralized electronic systems such as EDC, electronic data capture, to minimize errors associated with manual entry. Frequently cross-check adverse event reports and patient records for consistency. As for example, if a drug reality trial records an abnormal spike in a patient's drug concentration, monitors take immediate action by issuing an inquiry. The anomaly is found to be due to complete documentation of the patient's concurrent medication. Resolving such discrepancies ensures accurate representation of drug performance. Ensuring data integrity in drug reality trials also involves monitoring patient-reported outcomes and subjective feedback, which are often less structured than clinical data, but critical for evaluating real-world success. Safety monitoring. Safety is a priority in all clinical trials. Drug dimension trials focus on detecting safety signals under controlled conditions, while drug reality trials extend this monitoring to real-world populations where additional variables like polypharmacy and environmental factors may introduce new safety risks. And here is a summarized diagram on safety monitoring in clinical trials, dose adjustments for hepatic enzyme levels, protocols for tracking AEs, and SAEs, ongoing safety signal tracking, and their improved patient education protocols. And we have here the four areas divided by long-term strategy, real-world conditions, immediate response, and here controlled conditions. Approaches to safety monitoring establish robust protocols for tracking adverse events, AEs, and serious adverse events, SAEs, leverage pharmacovigilance PV systems to detect and respond to emerging safety issues, incorporate ongoing safety signal tracking even after the trial's conclusion to ensure long-term drug safety. As for example, a migraine drug in a drug dimension trial is found to increase hepatic enzyme levels in a subset of patients. Immediate dose adjustments are made to mitigate the risk. Later in a drug reality trial, safety monitoring highlights adherence issues in certain populations, promoting researchers to develop improved patient education protocols. Safety monitoring thus ensures that both controlled and real-world trial conditions prioritize the well-being of participants. The participants meaning or the patient's safety. Compliance. Compliance ensures that all trial components, ranging from ethical approvals to informed consent documentation, meet regulatory standards. Key areas to monitor confirm that all ethical and regulatory guidelines are followed at every site. Verify the accurate documentation of informed consent, including full disclosure of potential risks. As for example, a drug reality trial site fails to include details about potential allergic reactions in their informed consent forms, leading to intervention by monitors. Correcting these gaps prevents ethical violations and maintains trial integrity. Regular audits and timely corrective actions are critical for maintaining compliance across varied trial formats. And here we have a summarized diagram on ensuring compliance in clinical trials. We have ethical guidelines, verification, regular audits, informed consent documentation, and then corrective actions. Steps to effective monitoring. Step 1. Planning. The foundation of effective monitoring lies in a well-crafted plan. The risk profile and complexity of the trial guide the strategy for site visits, centralized monitoring, and interim analysis. As for example, for a high-risk oncology drug dimension trial, frequent site visits are planned to ensure adherence to dosing regimens and timely adverse events reporting. Steps to effective monitoring in clinical trials. Here a summarized diagram as starting from planning to execution to data review and then reporting. Step 2. Execution. Monitoring activities include real-time verification of protocol adherence, data consistency, and participant or patient safety. Both on-site monitoring and advanced remote tools play key roles in execution. As for example, during a drug reality trial, a monitor ensures that patient recruitment data aligns with inclusion criteria, preventing errors in trial population selection. Step 3. Data review. Regular data reviews help identify emerging trends, anomalies. Regular data reviews help identify emerging trends, anomalies, and opportunities for intervention. Advanced data systems should streamline this process with automated queries and pattern recognition tools. As for example, a clustering of post-dosage dizziness in a drug dimension trial initiates real-time analysis, revealing a need for pre-medication adjustments in high-risk patients. Step 4. Reporting. Comprehensive reporting creates a clear communication channel with stakeholders like sponsors, ethics boards, and regulators. Reports detail major deviations, outcomes, and any corrective and preventive actions. Example: a drug reality trial report notices significant adherence discrepancies in younger populations, leading to recommendations for improved monitoring mechanisms. Tools and techniques for monitoring. We'll start with monitoring software. Online platforms like Meditator streamline the capture and review of trial data, minimizing human error and improving efficiency, as for example, a drug dimension trial identifies incomplete lab entries through automated alerts, allowing immediate correction. And here is a summarized diagram on clinical trial monitoring techniques, root cause analysis, electronic data capture, data visualization, risk-based monitoring, and monitoring software. Then we have risk-based monitoring. After the monitoring software, we have risk-based monitoring, RBM, as RBM allocates resources to high-risk areas, improving efficiency and focusing efforts where they are needed most. As for example, a drug reality trial in rural regions prioritizes sites with high dropout rates for frequent oversight. Data visualization, dashboards, and graphs make it easier to interpret trends to identify outliers. As for example, interactive dashboards flag a pattern of dose reductions across multiple sites, allowing real-time investigation into potential causative factors. So, after monitoring software, risk-based monitoring, RBM data visualization, we have electronic data capture (EDC), as EDC systems ensure quick, accurate, and secure data entry and management. As for example, investigators upload patient vitals into an EDC system, enabling centralized safety monitoring. After that, we have also root cause analysis, and all these are tools and techniques for monitoring. So root cause analysis, this analytical tool uncovers the underlying reasons for deviations or anomalies in trial execution, as for example, low recruitment figures for a drug reality trial reveal investigator workload as the root cause, leading to targeted solutions. Examples of successful monitoring. Safety monitoring, as consistent tracking of SAEs, serious adverse events, during a drug dimension trial identifies early signs of drug-drug interaction risks, allowing protocol adjustments. And here's a summarized diagram on monitoring in clinical trials. We have safety monitoring, data integrity, compliance, protocol adherence, and risk-based approach. Then we have protocol adherence. A drug reality trial site with poor follow-up documentation is flagged early, enabling swift retraining and trial rescue. Data integrity, centralized query systems resolve discrepancies in lab values during both drug dimension and drug reality trials, preventing data delays. RBM or RB approach, risk-based approach, prioritizing oversight for high recruitment sites, reducing trial costs by 30% without jeopardizing data accuracy. We have compliance, real-time consent form validation prevents regulatory violations, ensuring trial integrity. Summary tables. Next will be we have tables to summarize the what we have covered through this lecture, definitions and significance, aspect drug dimension trials, and their drug reality trials. As definition, investigates drug safety and efficacy across varying therapeutic time points and doses. This is drug dimension trials, while in drug reality trials, assesses drug performance in real-world settings compared to clinical environments. We have the focus for each, the goal for each, the significance for each, and then an example on each. The drug dimension trials and drug reality trials. A table on key components of monitoring. Component. Description, how it's ensured, and then example. Components, we have protocol adherence, data integrity, safety monitoring, compliance, and real-world alignment. A table on steps to effective monitoring, step, objective, activity, focus areas, and then example. Steps, we have planning, execution, data review, reporting, and feedback loop. Table on tools and techniques for monitoring. Tool, technique, purpose, how it works, benefit, and example. The tools, techniques, we have monitoring software, risk-based monitoring, RBM, data visualization, EDC systems, and root cause analysis. Table on examples of successful monitoring, scenario, challenge faced, measures taken, outcome achieved. Scenarios, we have safety monitoring, protocol adherence, data integrity, risk-based monitoring, and regulatory compliance. Application table, scenarios, for each scenario, key principle applied, key actions, timeline, what to watch out for. Scenarios we have identifying reduced hypertension drug efficacy in real-world settings, resolving pharmacokinetic PK inaccuracies from deviated blood sampling times during dose escalation trials, querying unexplained anomalies like drug concentration spikes in CRFs, case report forms, adjusting migraine drug dosage after detecting increased hepatic enzyme levels, correcting consent forms, missing critical details about drug allergies, frequent monitoring of high-risk oncology site operations, investigating sudden dosage adjustment need trends across geographies through dashboards, flagging low recruitment rates in some sites and tracing issues to investigator engagement lapses, early SAE, serious adverse event reporting by clinical research associates, triggering drug interaction analysis and risk mitigation strategies in trials, preventing ethical violations during compliance checks by validating consent forms in real time, upscaling monitoring efforts on sites with higher pharmaceutical protocol deviation risks, implementing corrective actions for falsified patient follow-ups discovered during routine protocol adherence audits. Continuing with scenarios, adjusting resources based on centralized data analysis outcomes showing recruitment bottlenecks in multi-site global trials, using EDC systems to upload patient-reported outcomes directly and resolve flagged anomalies in oncology trial data, detecting and rectifying equipment failures at rural trial sites affecting real-time dosing data collection. And then we will be revising what we have covered through this lecture through some multiple choice questions or MCQs. Question 1, what is the main objective of drug dimension interaction trials? A, to study the effects of drugs in real world and controlled conditions. B, to investigate how drugs perform across varied doses and populations in controlled settings. C, to track patient adherence to drug therapies in everyday life. Or D, to compare drug pricing strategies across different markets. So from the four options, three of them real world. But the question really is about the main objective of drug dimension interaction trials. So because A applies to real world, same for C and D. So B would be the answer, as to investigate how drugs perform across varied doses and populations in controlled settings. As drug dimension interaction trials focus on understanding the safety and efficacy of drugs across different therapeutic time points, doses, and populations within controlled settings. Real-world conditions are evaluated in drug reality interaction trial, not drug dimension trials. Question 2, which of the following is an example of ensuring data integrity during drug trials? Is it A, checking for accurate and complete entries in case report forms, CRFs? B, identifying drug interactions in real-world dosage. C, ensuring proper use of consent forms across sites. Or D, monitoring adverse events AEs for immediate response. So A would be the most correct answer. As for checking for accurate and complete entries, data entries, and CRFs in case report forms, or the other terminology for it, or the name for it is EDC, electronic data capture. So A would be the answer, as in data integrity ensures that trial data is accurate and consistent, such as verifying complete and error-free CRF case report form entries. The other options represent safety monitoring and for D and compliance for C, but they are not example for direct data integrity measures. Question 3. What is a primary feature of risk-based monitoring (RBM) in a drug trials? A. Allocating equal resources to all sites of a trial. B. Focusing monitoring efforts on high-risk sites and activities. C. Completely eliminating monitoring at low-risk sites. Or D. Using real-time consent validation to ensure ethical compliance. So the most applicable for RBM, risk-based monitoring, as on the logic or idea of direct or guide or localize the efforts toward toward the high risk, meaning prioritizing according to the risk severity would be the most applicable one. B, as focusing monitoring efforts on high-risk sites and activities. So B is the answer. RBM emphasizes efficient use of resources by directing attention to high-risk sites and activities where deviations from protocol are more likely. Rather than applying uniform scrutiny, eliminating monitoring altogether at low-risk sites is not part of the RBM strategy. Question 4. How does data visualization enhance monitoring in trials? A, by immediately flagging trends or abnormalities in trial data. B, by automatically resolving data inconsistencies without human intervention. C, by replacing the need for safety monitoring protocols. Or D, by validating patient eligibility for trial participation. So the answer would be A, as data visualization helps to and supports in order to identify uh, certain data, certain trends. So A, by immediately flagging trends or abnormalities in trial data would be the answer. So A is the answer. So data visualization tools simplify the identification of trends like adverse events, AEs, or dose adjustments by displaying data points clearly. However, they do not replace standard monitoring protocols or human oversight. Question 5. Why is compliance monitoring critical in clinical trials? A, to ensure that informed consent processes are followed and ethical standards are met. B, to track dosage variations across study sites. C, to confirm the real-world applicability of trial outcomes. Or D, to improve interaction between monitors and trial stakeholders. So definitely, compliance monitoring is a critical because it A, to ensure that informed consent processes are followed and ethical standards are met. So A would be the answer. Compliance monitoring ensures all trial sites adhere to regulatory and ethical guidelines, including proper documentation of informed consent procedures. This safeguards participants' rights and ensures the study maintains its integrity. And here we come to end of this lecture. Thank you very much and goodbye.